Good afternoon, friends. In last lecture, I have discussed the different staining technique in that first, that is simple staining, and the different classification of staining technique, then simple staining and gram staining. Today, we will see also the gram staining and acid fast staining. In that first, gram positive. The gram-positive bacteria are those that are stained dark blue or violet by gram staining. Yes, what I am saying, the gram-positive bacteria are those that are stained dark blue or violet by gram staining. This is in contrast to gram-negative bacteria which cannot retain the crystal violet stain. Yes, keep in mind the difference. This is the contrast to gram-negative bacteria which cannot retain the crystal violet stain. It cannot retain the crystal violet stain. Instead of taking the counter stain, that is safranin or fuchsin, then and appearing the red or pink color, where the gram positive organisms are able to retain the crystal violet stain because of high amount of peptidoglycan which is present in cell wall. Yes, what I am saying, the gram positive bacteria which are capable or which are able to retain the crystal violet stain. Because of why this bacteria retain the crystal violet stain? Because it contain these gram-positive bacteria which contain the high amount of peptidoglycan which is present in cell wall. Then the gram-positive cell wall is typically like the outer membrane and found in gram-negative bacteria. Then here we can see the structure of gram-positive bacteria. In that, yes, we can see here the tetroic acid, then embedded protein, then peptide bond and cell membrane, and peptidoglycan cell wall. Here we can see the structure of gram-positive bacteria. In that, what you are seeing, the tetroic acid, then peptidoglycan cell wall. It means the cell wall which contain peptidoglycan, then embedded proteins, peptide bonds, and cell membrane. The gram-positive bacteria are characterized by what? It characterized by having as a part of their cell wall the structure peptidoglycan as well as polysaccharide and the tetroic acid. Yes, it shows presence of what? Peptidoglycan as well as polysaccharides and tetroic acid. The peptidoglycans which are sometimes are also the peptidoglycan which are also called as murine. Yes, keep in mind, it is important for MCQs. This peptidoglycan which is or is called, sometimes is called as what? Murine. These are heteropolymers of glycan strand. Yes, murine. These are the heteropolymers of glycan strands and which are cross-linked through short peptides. Yes, which are cross-linked through the short peptides. Then we will see the what are gram-negative bacteria. Gram-negative bacteria are those bacteria that do not retain the crystal violet. Yes, these are the bacteria which does not retain the crystal violet dye in the gram staining protocol. Yes, what I am saying, these are the bacteria or those bacteria that does not retain the crystal violet dye in the gram staining protocol. In gram stain test, a counter stain that is commonly safranin is used as a counter stain is added after the crystal violet, the coloring all gram-negative bacteria with red or pink color. Then this test itself is useful for classifying two different types of bacteria which are based on structural difference of their cell walls. And on another way or other hand, the gram-positive bacteria will retain the crystal violet dye when washed in decolorizing solution. Then here we can see the structure of gram-negative bacteria which shows presence of lipopolysaccharide then protein, cell membrane, and peptidoglycan. Almost the gram stain preparation, the gram negative organism will appear red or pink color because they are counter stained. Why it shows pink color? Because it is counter stained due to the presence of higher lipid content. Yes, due to the presence of higher lipid content after alcohol treatment, the porosity of the cell wall it increases. The crystal hence the CVI complex, that is crystal violet iodine complex, it can pass through. Thus, the primary stain is not retained. Here we can see the structure of gram negative bacteria. Yes, gram negative bacteria also in contrast to most gram positive bacteria, the gram negative bacteria have only a few layers of peptidoglycan and a secondary cell membrane which is made primarily of lipopolysaccharide. Yes, in contrast, the most gram positive bacteria and gram negative bacteria have only a few layers of peptidoglycan and a secondary cell membrane which is made up of primarily the lipopolysaccharide. Then these are the few questions which are generally asked: principle, then different methods, then we will, so this is all about gram staining. We will see next staining technique, that is acid fast staining. Yes, the acid fast staining in modification of Enlage in 1882. This method is also known as Ziel-Nelson stain or Ziel-Nelson staining. Keep in mind, this acid fast staining is also called as what? It is also called as Ziel-Nelson stain. It is used for bacilli belonging to the genus Mycobacterium, especially for Mycobacterium tuberculosis, then Mycobacterium leprae, and also for Nocardia. Yes, what I am saying, this acid fast staining is a modification of Enlage in 1882. Enlage method in the year 1882. This staining is also called as what? Ziel-Nelson stain or Ziel-Nelson staining. And this acid fast staining or Ziel-Nelson staining is used for what? It is used for bacilli belonging to the genus Mycobacterium. And this staining is especially used for what? The Mycobacterium tuberculosis, then Mycobacterium leprae, and also the Nocardia. Then this acid fast staining or the acid fastness of this acid fast bacilli is attributed to the presence of unsaponificable wax fraction, and which is called as mycolic acid, and which is present in their cell wall of bacteria. Here's what I am saying. This acid fastness of this acid fast bacilli, which is attributed in presence of what? In presence of unsaponificable wax fraction, and which is called as mycolic acid in their cell wall. Then we will see the basic requirements. First, the primary and modern staining with strong carbol fusing, that is red, then decolorization with acid alcohol, then counter stain with methylene blue. Yes, for acid fast staining or Ziel-Nelson staining, the primary stain and modern stain, it is strong carbol fusing. Then for decolorization, use of acid and alcohol, and for counter stain there is use of methylene blue. Keep in mind these stains. What I am saying, the primary and modern staining, what we use for acid staining, that is strong carbol fusing. Then for decolorization, yes, use of acid alcohol, and for counter stain with or by the use of methylene blue. Next we will see the procedure for acid fast staining. First, make a smear, then air dry, then heat fixation. As I already discussed, what is heat fixation, what is preparation of smear in detailed procedure. Then first make a smear, then air dry, then heat fix, then fluid the smear with carbol fusion, that is primary stain, then steam for 5 minutes, then add more of the stain as needed, then cool the slide for 5 minutes, then after cooling wash the slide with distilled water, then fluid with acid alcohol, that is decolorizer. So what is the use of acid alcohol? That is, it is used as a decolorizer, and float the slide with acid and alcohol and leave the slide for 15 seconds. Then tilt the slide 45 degrees Celsius over the sink and add acid alcohol dropwise until the red color stops streaming from the smear. Yes, till what? The add the acid alcohol dropwise until the red color stop the streaming from the smear. Then rinse the slide with distilled water, then add Loeffler methylene blue stain, that is, it act as a what? This methylene blue, it act as a counter stain. Then after that, leave the stain on the smear for 15 to 20 seconds. Then after that, rinse the slide and dry it, that is natural drying. Then and it is observed under oil immersion objective, that is 100x objective. Then what will be the result or observation for acid fast staining? This is all about procedure. I am repeating the procedure. First, make a smear, then air dry and then heat fixation, then float the smear with carbol fusion stain, that is your primary stain, then steam for 5 minutes and add more of the stain as a needed, then cool the slide for cool the slide for 5 minutes and wash with distilled water, then fluid with acid alcohol, that is your decolorizer, and leave the slide for 15 seconds, then tilt the slide 45 degrees Celsius over the sink and add the acid alcohol, that is decolorizer. Add the acid alcohol dropwise until the red color stops streaming from the smear, then rinse with distilled water, then add Loeffler methylene blue, that is, it acts as a counter stain. Add the methylene blue, then leave the slide or leave the stain for or the smear for 15 to 20 seconds, then rinse the slide until it dry, and at the last observe the slide under microscope. Then what will be the result or what will be the observation for this acid fast staining? The stained smear are contain pink color slender rod shaped structure as seen with the curved ends, and the smear is positive for acid fast bacilli. Here we can see the structure or observation. The acid fast bacteria, Mycobacterium gordoni, and non-acid fast bacteria, Staphylococcus epidermidis. Yes, for acid fast bacteria it is Mycobacterium gordoni, and non-acid fast bacteria Staphylococcus epidermidis. Yes, this is all about the acid fast staining. Then we will see the capsule staining. So what is capsule staining? The capsule serve as a protective material for slowing down or preventing the prevention of chemical and body juices. Yes, capsule staining, the capsule it serve as what? It serves as a protective material by slowing down or preventing the prevention of chemical and the body juices. So we will see the principle of this capsule staining. First, the chemically the capsular material is a what? It is a polysaccharide. It is glycoprotein or polypeptide. This capsule staining, it is a chemically capsular material which is a polysaccharide. It is a glycoprotein or polypeptide. Then this capsule staining is more difficult than other type of differential staining. Yes, keep in mind this staining is very difficult than the other type of differential staining because his capsular material are water soluble and may be dislodged, dislodged and removed with vigorous washing. Yes. Why it is difficult? Because this capsule it is, or this capsular material it is water soluble, and because after staining there is chances of it may be washes during the different stain washing. So therefore it is difficult than the other staining. Why it is difficult? Because this capsular material it is water soluble, and during staining procedure we use the different stain, and after staining we will use at UV staining the slide will be washed. Therefore there may be the chances of dislodged and removed with vigorous washing. Then this capsule is non-ionic, so that the dyes commonly used will not be bind to it. The second reason, that is, what is second reason? This capsule is non-ionic, so that this dyes is commonly, the dyes commonly used will not be bind to it. Two dyes are the two dyes, one acidic and one for. Then we will see the procedure. For positive staining of smear, make a smear from colony of pneumonia on a clean grease free slide or glass slide, and allow to dry in air. Then float the smear with crystal violet and allow it to stand for 5 to 7 minutes. Then wash the smear with 20% of copper sulfate solution and dry it. Then wash the smear with 20% copper sulfate solution and then dry it. Then observe the smear first under low power objective, that is 10x objective, and then under the slide and then observe the slide under 100x objective, that is oil immersion objective. In the culture smear, the capsule is seen as a light blue in contrast to the deep purple color of the cell. In color smear, the capsule is seen as a light blue in contrast to the deep purple color of the cell. Yes, what is procedure for positive staining of smear? Make a smear from a colony of Staphylococcus pneumoniae on a clean grease-free glass slide, then allow to air dry. Then float the smear with crystal violet and allow it to dry, or allow to stand or allow to stand for 5 to 7 minutes. Then wash the smear with 20% of copper sulfate solution and dry it. Then observe the smear under, firstly the observe the slide under 10x microscope, 10x objective, that is low power objective, and after that then observe the slide under oil immersion objective, that is 100x objective. In the culture smear, the capsule is seen as a light blue in contrast to the deep purple color of the cell. This is all about what? Smearing of that is positive staining of smear. Then next we will see the negative staining of smears. Take a clean grease-free glass slide. Then put a large loopful of undiluted Indian ink on the slide. Put a large loopful of undiluted Indian ink on the slide. Then add the small loop of this. Then add the small loopful of liquid bacterial culture to this Indian ink and emulsify. Then take a clean grease-free cover slip and place on this ink drop and press it down so that the film become very thin and thus pale in color. Then this then this slide observe under wet. Then observe the wet film under high power objective, that is 40x, and this capsule in negative staining method is seen as a clear refractile around the organism against the black. Yes, what is the procedure for negative staining of smear? First, Take a clean, grease-free glass slide. Then put a large loopful of undiluted Indian ink on the slide. Then add a small loopful of put large loopful of undiluted Indian ink on the slide. Then add the small loopful of liquid bacterial culture. Then add the small loopful of liquid bacterial culture to the Indian ink and then emulsify. Then next, take a clean grease-free or cover slip. Take a clean grease-free cover slip and place on the ink drop and press it down so that the film become very thin and thus it is pale in color. Then observe the wet film under high objective or the high power objective, that is 40x, and this capsule in negative staining method is seen as a clear refractile around the organism against the black. Here we can see the capsule staining. Here we can see this is background, then it is capsule and it is rods. Here we can see the clear image. It is background, then it is capsules and it is rod. Then we will see the endospore staining. Yes, this is all about the capsule staining. Here we can see the background, then it is capsules and it is rods. Then we will see the endospore staining. The spores are highly resistant in active forms. Yes, what I am saying, the spores are highly resistant in active forms. The morphology of bacterial endospore is best observed in unstained wet film under the phase contrast microscope, where they appear as a large refractile oval or spherical bodies within the bacterial mother cells or else free from the bacteria. Yes, different staining are available for staining of spores. The spores are highly resistant to, it means the spores are highly resistant in inactive forms. The morphology of bacterial endospores is best observed in unstained wet films under the phase contrast microscopy, where they appear as large refractile oval spherical bodies within the bacterial mother cell or else free from the bacteria. The different stainings are available for staining of spores. We will see the procedure. Films are dried and fixed with minimal flaming. Then place the slide over a beaker of boiling water. Place the slide over a beaker of boiling water, resting it on the rim with bacterial film uppermost. Then, when within several seconds the large droplet have condensed on the under the side of slide, then it is flooded with 5% of aqueous solution of malachite green and leave it for 1 minute while the water continues boil. Then wash it with cold water. Then treat with 0.5% of safranin and 0.05% of basic fusion for 30 seconds. Then dry it. This method colors the spore green and vegetative bacilla red, and liquid granules are unstained. Here this method colors the spores are green, then vegetative cells are red, and these liquid granules are unstained. Here we can see the endospores. Then we will see the summary of these staining techniques. It means after watching this video in the sense there are 3 to 4 videos, that is from introduction of staining techniques. We are able to explain the staining technique which is used in microscopy. Yes, we will see the summary. Staining is a technique used in microscopy to enhance the contrast in the microscopic image. Then stain is a substance that adhere to the cell and it gives or giving the cell color. Then stain are classified as a simple stain, differential stain and spatial stain. Then gram staining is used to differentiate the bacterial species as gram positive and gram negative based upon the chemical and physical properties of the cell wall. Then the acid fast staining, it is also called as what? Ziehl-Neelsen staining. It divides the bacteria into acid fast and non-acid fast, and this is used in diagnosis of tuberculosis and leprosis. This is the summary of this staining technique. This is all about the all staining techniques. I will stop here.